reflect the cause or symptoms of an underlying brain condition that is of interest now. And answering these questions is critical to harnessing the intestinal microbiota as a tool for ameliorating or preventing brain disorders and determining potential links with metabolic and cardiovascular diseases, and for developing nutritional and therapeutic strategies that support and strengthen the brain health of the individual. So now continuing to this topic, and let me start my lecture. Yeah. Yes. So I would like to portray this gut-brain axis as a dance between the gut and the brain. Suppose if you imagine two dancers in perfect sync with each other. 1 minute, sorry. Yeah. So it's as if one senses the other's movements before they even happen, and they adapt and respond instinctively, and their coordination is precise. So a dance is graceful when there is just the right degree of harmony and balance in movement. So the gut doesn't just react; it can sense, remember, signal, and even anticipate. So it does so through a physiological balance with the brain and within itself also. So the gut's microbiome is a beautiful little stage with its own little dancers, which are called the microbiota, and that exist in a bidirectional relationship with the brain. So not only does it allow the brain to, the gut to talk to the brain, it also allows it to listen. And as it responds to various neuronal, hormonal, immune, and metabolic cues, it undergoes frequent shifts in its composition. So in this lecture, we will explore this vast and sophisticated choreography of neurons, hormones, immune signals, and microbial metabolites. So instead of seeing the brain as a sole command center that the gut merely responds to, we will see how they are in active dialogue with one another. So, so outline of this topic. So we are divided it as the dancers, the stage, and the dance per se. So the dancers are the enteric nervous system and the vagus nerve, and having the afferent and efferent signaling. The stage is the microbiome, then the intestinal physiology related with it, and the changes in its composition, the permeability, the barrier structure. And the dance itself is the gut-brain axis with its neuronal, endocrinal, metabolic, and immune signalings. So all diseases begin in the gut, given by Hippocrates. So coming into the history, in the past decades, a new player has emerged as a key regulator of the gut-brain axis, the trillions of microbes within the gut, the microbiota. So 5 separate lines of evidence converge to establish this. So first is the studies in germ-free animals showed that the brain is affected in the absence of microbiota. Second, the animals given specific strains of bacteria had alterations in behavior, and human studies of such strains confirm the potential translatability of such findings. And the third, population studies given by most notably in Volkerton in Canada demonstrated alterations in the gut-brain symptoms, and these findings were also echoed in animal studies where low-level infections altered behavior. And fourthly, the preclinical studies with antibiotic administration, either in early life or adulthood, have shown long-lasting effects on brain, spinal cord, and the enteric nervous system. And finally, these data synergized with a long-known clinical situation that hepatic encephalopathy could be broadly treated by targeting a microbiota with antibiotics in humans. So once it was understood that our common self friends in the gut could effectively communicate with our brain, a rush of studies sought to understand the intricate processes involved. So this begins the concept of microbiota gut-brain axis thus emerged. Now, And the phrase microbiota gut-brain axis is often mistakenly interpreted as one-way street, that is communication principally in the direction of gut to brain. While numerous reviews have emphasized the bidirectional nature of gut to brain communication and the consideration of microorganisms as neurochemical producers that also possess cognate high affinity receptors, means that the microbiota is responsive to signals from the brain to the gut and as such can alter its function and composition in response to host originated neurochemical signals. So this is a bidirectional microbiota gut-brain axis showing 4 different communication pathways: the microbial factors, the gut hormones, the cytokines, and the sensory neurons. Signal from the gut to the brain where they can modify cerebral function and behavior. And the 2 pathways are the autonomic neurons and the neuroendocrine factors or the outputs, which this signal from the brain to the gut. So again, this brain, so the gut microbiota, gut mucosa, the gut immune system, the gut-brain axis, and the brain per se, all these are in communication with each other. Now coming to the, so now just so this is the diagram showing you the gut-brain axis, wherein all the bidirectional involvement is taking place, involving the neuronal messages, endocrinal messages, immune messages through the either through the systemic circulation or through the blood-brain barrier happening. And here if you see, we see just I want to introduce the dancers, what we were talking about, the enteric nervous system being the second brain and the other dancer being the afferent, the vagus nerve here. So in 1899, that is 116 years ago, Bayliss and Starling proposed the law of the intestine. So putting forward the idea that gut innervation seems to work differently from other parts of the body. And local stimulation of the gut produces excitation above and inhibition below the excited spot. And these effects are dependent on the activity of the local nervous mechanism. And unlike skeletal muscles, which rely on direct input from the central nervous system, we now know that the wall of the gut is home to a complex neuronal circuitry. So the gut must have, for that, the gut must have some way to sense the stimulation. The gut can sense the stimulation through chemical signals or detecting distension, transmitting these messages either upstream or transmitting the message downstream. And remarkably, all of this has to occur within the walls of the gut, and it does thanks to the enteric nervous system. So before going to this, wish one more thing important, cool potential direction for the session is interesting to us. That is, we should know that the developmental connection between the enteric nervous system and the central nervous system, how they originate from the same embryonic neural crystals is important. And gut is not just connected to the brain, but evolutionary related to it. And the stunning complexity of the enteric nervous system that it produces more than 30 neurotransmitters, including serotonin, dopamine, acetylcholine, and nitric oxide, making it more complex than the spinal cord. Then coming to the vagus nerve, which serves as the primary bidirectional communication pathway between the gut and brain, playing a crucial role in regulating digestion, immune response, and overall homeostasis. And most important thing we should know is its fibers are predominantly, that is approximately 80 to 90% are afferent, transmitting information from the gut to the brain. So the vagus nerve effectively functions as a sensory conduit. And how does this afferent signaling occur? So afferent vagal fibers in the gut mucosa layers can detect a wide range of stimuli. For example, the mechanical signals or chemical signals, immune signals involving the stretch receptors, nutrient compositions, then pH changes or any inflammatory cytokines. And other afferent signaling is via the lamina propria. So the connective tissue layer beneath the gut epithelium, which is particularly rich in immune cells and neuroendocrine signaling molecules. And it acts as an interface for vagal sensory input. So this is where the vagal afferents are exposed to gut-derived hormones like cholecystokinin, then serotonin, etc. And also we know that these relay the nutritional status to the brain. So these hormonal and neural outputs have the ability to influence the gastrointestinal targets such as immune cells, enteric smooth muscles, enteric neurons, and enteroendocrine cells. So what is this neuronal signaling actually? So the neurons that innervate the GI tract can be divided into the primary extrinsic, that is the vagal and spinal afferents, or primary intrinsic afferent neurons, with the intrinsic neurons being much more abundant. So both the extrinsic and intrinsic neurons respond to, as I told you, the mechanical, chemical, or immune signal stimuli. And the luminal signals do not influence afferent nerve terminals directly, but they act through multiple reflex loops to optimize the gut function. And there are differences in both the extrinsic and intrinsic neurons with regards to the importance of these reflex loops depending on their location. So the stomach is predominantly governed by vagovagal reflexes. Thus, signals arising from extrinsic and intrinsic neurons are relatively weak. In the intestine, the intrinsic primary afferent neurons and enteric motor neurons are important for intestinal function. Efferents are much stronger in the intestine, which are reliant on these signals. The intrinsic pathway afferents feed and regulate signals relating to secretion, propulsion of food and blood flow indirectly to the central nervous system through specific myenteric ganglia, which receive input from these neurons as well as extrinsic efferents. And this information is relayed to the spinal, mesenteric, and supraspinal reflex loops that span much larger distances in the ball. So this is about the vagus or the enteric nervous system per se. Now coming to some important terms, what we should know, that is the microbiome. So the totality of microbial genomes present within a given environment, that is the gut, that is called a microbiome. The microbiota, the actual microbial organisms themselves, along with their function, metabolic activity, and interaction with the host. Then the final term, that is dysbiosis. Any disruption in this microbial ecosystem, which has an impact on host function, that is the dysbiosis. So how does the microbiota influence intestinal physiology? So over 2000 bacterium species for about have been identified so far, and 93% of them have been classified within 4 dominant phyla. So, for example, they are the proteobacteria, firmicutes, actinobacteria, and bacteroidetes. So, this microbiome is a diverse microbial ecosystem within us that includes not only bacteria, but also viruses, fungi, protozoa, and all of which interact dynamically with the host and with each other. When we say microbiome, we are primarily referring to the bacterial community residing within gut. And there is also the mycobiome that is involving the fungi, which is largely Candida and the Saccharomyces. And the microbiome makes up less than 0.01% of the total microbiome, but there's increasing interest in how these fungi also can influence bacteria as well. Therefore, it's not just bacteria that we say microbiome, but today we are mostly going to focus on this bacterial microbiome. Then, so what are the key physiological roles of the gut microbiota if we take into consideration? The majority reside in the distal wall, where they aid in breaking down dietary fibers and complex carbohydrates. And fermentation of these fibers produces short chain fatty acids, and which we will explore in detail soon, next in the next slides. And right from birth, gut microbes play a role in educating the mucosal immune system. And microbiota-derived metabolites interact with the dendritic cells, T cells, and the regulatory cytokines. And also the microbiota's effects are not just restricted to the gut. They also extend to the brain, liver, and cardiovascular system through metabolic and immune signaling pathways. And the gut microbiota also play a critical role in maintaining the blood-brain barrier integrity through the modulation of tight junction proteins. So what alters the gut microbiota composition over time? So there are different internal factors and external factors. Internal factors like changes in mucin production, mucus secretion, changes in the secretion of antimicrobial peptides, defensins, secretory immunoglobulin A, then bile acid composition, psychological stress, any conditions that alter are, for example, the gastrointestinal functional disorders, such as IBS, inflammatory bowel syndrome, then chronic inflammation or activation of mucosal immune system. And coming to the external or the environmental factors like exposure to antimicrobial drugs or using antibiotic for a prolonged time, then dietary changes, infection, surgical procedures involving GI tract, and drugs that alter intestinal physiology, example, the proton pump inhibitors, antidiarrheal agents, etc. And also alterations in the biodome and microbiome also can indirectly influence the bacterial composition. So how does the microbiota influence intestinal permeability? So gut microbiota plays a very key role in maintaining intestinal permeability, which is essential for nutrient absorption by preventing the entry of harmful antigens. So what are the different microbial conditions here? First one is the germ-free conditions where the permeability is not at all applicable, but effect on gut barrier, there is immune mature system, so immature tight epithelial barrier. Coming to normal microbiota, it's normal selective permeability happening and maturation of immune defenses, sorry. Then dysbiosis or infection, that is where there is increased permeability and here the barrier function is impaired and giving various degrees of inflammation. Next question is, how does the microbiota influence intestinal barrier structure and function? So before this, we should note that when the gut and brain are less tightly connected, there is increased intestinal permeability, which is also called the leaky gut, which can cause an excessive inflammatory response. So now, how does the microbiota influence intestinal barrier structure? We know that the intestinal barrier serves as a selectively permeable barrier that allows nutrient and water absorption while preventing the translocation of pathogens, toxins, and inflammatory antigens. So the gut microbiota plays a fundamental role in regulating each structural and functional component of this barrier, which includes the lumen, the mucosal barrier, the epithelial cells, the tight junctions, lamina propria, and the innate immune cells. So the extracellular environment that contains the gut lumen. Okay. So the extracellular elements are containing the gut lumen, which contains the microbiota, that is the bacteria. Okay. Microbiota, the bacteria, viruses, fungi, archaea, or any protozoa. And the dietary components, that is the fiber or proteins, lipids, or micronutrients. And the metabolites, they are the short chain fatty acids, bile acids, and the antimicrobial peptides. So these microbiota modulate gut barrier integrity by producing these bioactive compounds or metabolites. So, and then the next layer is the mucosal layer, which promotes mucin production and regulates mucin glycosylation, okay, and also prevents pathogenic overgrowth. So here we should know that this mucus has potential antimicrobial activity because it contains antimicrobial peptides, AMPs, such as alpha and beta defensins that are secreted by the intestinal epithelial cell lineages, such as Paneth cells, for example, the Paneth cells, and secretory immunoglobulin A produced by the plasma cells located in the lamina propria. And this secretory immunoglobulin A is the most abundant immunoglobulin of bacterial of this mucosal surfaces, and it plays a significant role in maintaining the barrier function within the intestinal mucus by binding to the bacteria in the gut lumen with some level of specificity and even preventing microbial invasion. The next is the most important physical barrier at the intestinal mucosal surface. Underneath the mucous layer is the intestinal epithelium. So these cells form a continuous and polarized epithelial barrier with several cellular lineages. The absorptive cells, the goblet cells, the Paneth cells, the enteroendocrine cells, and the tuft cells. So each cell type has specific functions. For example, both goblet cells and the Paneth cells help to fortify the mucosal barrier through the production of mucus and the antimicrobial proteins. And the substances can pass through this barrier via two general pathways, that is the paracellular and the transcellular pathways. And these intercellular complexes are also under neurohumoral control, such as from vasoactive intestinal peptide and cholinergic neurons from the submucosal plexus. And located primarily below the intestinal epithelium, some component situated in between is the mucosal barrier, the immunological barrier of intestinal mucosa, known as the mucosal immune system. This is known as the mucosal immune system, which is the largest immune organ in the human body, consisting of these different cells like plasma cells, dendritic cells, macrophages, etc. Then, so this is a complex and interconnected array of various immune cell populations, maintains intestinal mucosal immune homeostasis by balancing immune activation to protect against microbial invasion. And the general populations of this intestinal immune effector system include the T effector cells, regulatory cells, B cells, dendritic cells, components of the innate immune system, such as macrophages, mast cells, okay, and even neutrophils. And an important and more recently described cell population is the innate lymphoid cell, which belong to the lymphoid lineage, but lack antigen-specific T and B cell receptors. So interaction between all these components of the intestinal mucosal barrier is complex, but is well orchestrated to maintain a homeostatic state between the host and its environment at the mucosal interface. For example, these innate lymphoid cells are particularly important in the maintenance of the intestinal mucosal barrier through the secretion of interleukins 22 cytokine, which promotes both enhanced mucus production via the goblet cells, okay, as well as the antimicrobial proteins. And both of these are important components of the intestinal mucosal barrier. And next is the tight junctions. So the junctional complexes in the intestinal epithelium. So these molecules can cross the intestinal epithelium through monolayer through the intercellular spaces, either paracellular way or the transcellular way. And the junction complexes involved in paracellular pathway include the tight junctions, adherent junctions, gap junctions, and desmosomes. And the tight junctions are composed of these transmembrane proteins such as claudins, occludin, junctional adhesion molecules, and zonular occludin proteins. And these zonular occludin proteins or occludins act as scaffold proteins connecting claudins and occludin to the cytoskeletal actin. So this is the host genetic factors, and whenever the microbiota are increased, so there's loss of this epithelial barrier integrity, causing tissue destruction and mucosal inflammation due to uncontrolled immune response. So there's disruption of these cells taking place. Now let us move on to the dance, or what we call the gut-brain axis. So for the gut and brain to dance together, how does the gut talk to the brain? And how does the brain talk to the gut? How does the gut listen to when the brain talks or responds? So all this is via this bidirectional gut-brain axis. So the ability of the brain to influence the intestinal microbiota, or the ability of the microbiota to influence the brain and behavior. So via different neuronal, endocrinal, immune, metabolic mechanisms. So involving the HPA axis, the vagus, and also the bacterial metabolites and the neurotransmitters. So this gut-brain axis, first a bidirectional communication involving all these signaling mechanisms. And to just outline the neural, if you see, it is through the vagus nerve, the afferent from the gut to the brain, efferent from the brain to the gut, and also the enteric nervous system, which is also called the second brain. Now coming to the neural signals, if you see, the afferent part of this gut to the brain system is important in the regulation of the appetite control, then perception of pain, and in monitoring by the brain of the inflammatory and immunological status. So, if you see the neuroendocrinal signaling, for example, in the gut microbiota brain communication, the neuroendocrine level primarily includes the HPA axis, which plays a key role in stress response and is also one of the main components of the gut-brain axis. The HPA axis begins in the paraventricular nucleus of the hypothalamus, where the corticotrophin releasing factor is synthesized. And this stimulates the pituitary gland to produce the adrenocorticotropic hormone, which leads to the release of glucocorticoids, that is cortisol and corticosterone from the adrenal cortex. In addition, this corticotrophin releasing factor stimulates the locus coeruleus to synthesize catecholamines, thus leading to an increase in the non-adrenergic activity of the brain. And there is evidence that the gut microbiota develops in parallel with the HPA axis, and they are in constant communication. This is the communication. And this two-way communication between the gut microbiota and the HPA axis is increasingly emphasized. And also the abnormal formation of this HPA axis during brain development may affect the microbial colonization and visceral sensitivity. So any abnormality of this, the HPA axis being activated by the interleukin 6. So this interleukin 6 downregulates the glucocorticoid receptors to produce this cortisol. So increased cortisol leading to an increase in the gut permeability, causing leaky gut. And this derives a pro-inflammatory state as evidenced by increased levels of circulating tumor necrosis factor alpha, then interferons alpha, gamma, and interleukin 6. And this interleukin 6 activates the HPA axis and again down regulates the glucocorticoid receptors, removing the negative feedback loop, resulting in a hypersensitive HPA axis. Then coming to the vagus. So the vagus, as we know, vagal efferents are distributed in all the layers of the gastrointestinal wall, but do not penetrate the epithelial layer into the lumen of the intestine. And the efferent endings of the vagus nerve have been divided into 3 subtypes. So the afferent endings are located in the apices of the intestinal villi, and also the endings around the intestinal glands or the crypts, forming the cryptic efferents, and the efferent endings along the antral glands of the stomach. And all of these vagal endings are both chemosensitive and mechanosensitive. And these vagal nerve chemoreceptors are presumed to be involved in communication between the gut microbiota and the brain by detecting the small chain fatty acids. And these vagal efferent nerves extend into the myelinic plexus, release different neurotransmitters to regulate the smooth muscle contraction and the gastrointestinal movements. And this the information which is detected when there is an interface, the vagal efferents transmit these signals via their cell bodies, which are located in the nodose ganglion. And they synapse in the nucleus tractus solitarius of the relay center of the brain, which is the medulla oblongata. And what happens within this nucleus tractus solitarius? The signals undergo initial processing before being transmitted to various brain regions. That is, for example, the paraventricular nucleus of the hypothalamus for homeostasis and autonomic function. The periaqueductal gray matter, aqueductal gray, sorry, for modulating pain perception. Here, pain perception and also autonomic response. Then, if you see, the amygdala and the insula plays a role in emotional and cognitive responses to gut-derived signals. And this nucleus tractus solitarius also projects to the dorsal motor nucleus of the vagus nerve, which forms a reflex loop that outputs into the gastrointestinal tract. It allows for rapid adjustment-based responses in either gastric motility, secretion, or inflammation. Then, how does the gut? Next is how does the gut sense these changes? So the gut senses these changes via the enteroendocrine cells located in the intestinal epithelium, which detect luminal contents and secrete hormones like cholecystokinin, GLP-1, and polypeptide YY, or peptide YY, sorry, and that signal the brain via the vagus nerve. And pattern recognition receptors, PRRs. These include toll-like receptors and nod-like receptors, which detect microbial-derived molecules such as lipopolysaccharides and peptidoglycans. And immune cells in the lamina propria, dendritic cells, and macrophages also process the microbial signals and secrete cytokines. And neural sensory pathways, that is the vagus and the spinal efferents. Now coming to the intestinal barrier, if you see the gut bacteria metabolize dietary fibers and complex carbohydrate, producing the short chain fatty acids, which are the key molecules in gut-brain communication. And the three principal small chain, short fatty acids are short chain fatty acids modulate the gut. And these enteroendocrine cells are the taste cells of the gut, are specialized endoderm-derived epithelial cells, which differentiate from the pluripotent stem cells at the base of the intestinal crypt villus axis. They migrate up to the crypt villus axis, and the distribution of these EECs in the villi and crypts, and they are interspersed between the non-endocrine cells. And interplay of different factors occur, including the self-surface protein, that is notch, and the basic helix loop helix transcription factor family, which control the terminal cell differentiation. And depending on their individual morphology and position of in the GI mucosa, these cells are divided into open type and they're divided into open type and closed type of EECs. The open type EECs directly detect luminal contents to the microvilli reaching the lumen, whereas the closed types are believed to be activated by luminal content indirectly, either through neural or humoral pathways. So both these types of accumulate their secretory products in cytoplasmic granules and release them by exocytosis at the basolateral membrane upon either mechanical or chemical or neural stimulation. And the secretory products are mainly the peptides or hormones, which can act locally in a paracrine manner, activating other enteroendocrine cells and other cell types in the mucosa. And they reach distant targets through the release in the bloodstream or act directly on the nerve endings close to the site of release. And these hormones released by the enteroendocrine cells upon stimulation act on receptors located on the vagal or spinal neurons and influence neuronal pathways in a bidirectional manner. Now, so the lipopolysaccharides are pro-inflammatory antitoxin, that is component of the outer membrane of the gram-negative bacteria. And these activate the inflammatory response, which in turn increases indolamine dioxygenase. So activating the pathway. Now, if you see the different pathways, for example, the small short chain fatty acids, which promote the secretion of gut hormones from the enteroendocrine cells and indirectly affects the brain via the systemic circulation and vagal pathways, already I have mentioned. And in the brain, these small short chain fatty acids also increase satiety, microglia, neurogenesis, and decrease blood-brain barrier permeability. And the talking after sensing is happening through these bacterial mechanisms to generate this bioactive short chain fatty acids. So what is the importance of these short chain fatty acids? So these influence the gut brain communication. They're released because of so these are like three different types, that is acetate, butyrate, and propionate. These short chain fatty acids in circulation are very short-lived with low concentrations in the bloodstream. And they're also coupled to different types of receptors. For example, the free fatty acid receptor 2 and 3 and GPRs. And they influence the intestinal mucosal immunity and barrier integrity and function. And also the interaction with the receptors on the enteroendocrine cells promote indirect signaling to the brain via the systemic circulation, all vagal pathways by inducing the secretion of gut hormones like GLP-1, peptide YY, GABA, and serotonin. And also they have different functions like regulation of liver mitochondrial function, increased insulin secretion, then whole body energy, homeostasis. And their action peripherally is influence systemic inflammation, mainly by inducing T regulatory cells called TREG differentiation and by regulating the secretion of interleukins. And they can also cross the blood-brain barrier via the monocarboxylate transporters located on endothelial cells and influence the integrity by upregulating the expression of tight junction proteins. And their effect on the central nervous system, they influence neuroinflammation by affecting glial cell morphology by modulating the levels of the different neurotrophic factors, increasing the neurogenesis, and contributing to the biosynthesis of serotonin, and also improving the neuronal homeostatic function. So indirectly or directly, they affect the emotion, cognition, and pathophysiology of different brain disorders. So these are the different functions of the short chain fatty acids acting through the microglia or astrocytes and the blood barrier integrity. Then another important thing is the gut-liver axis. The gut microbiota and the metabolites, as well as some pro-inflammatory products, can pass through the intestinal barrier and enter the liver through the portal system. And in the liver, they promote or inhibit the progression of non-alcoholic fatty liver disease through different mechanisms. And also can regulate intestinal function and gut microbiota balance through the bile acid circulation, which is an important enterohepatic circulation in regulating this. So that is the role of gut-liver axis. And next, under normal conditions, we see about 90% of the assumed tryptophan is catabolized and transformed into kynurenine through the kynurenine pathway. And while approximately 3% is metabolized into serotonin. And the remaining is degraded by the gut microbiota to produce indole and its derivatives. And this kynurenine pathway represents, therefore, the main tryptophan degradation pathway, leading to the formation of a large number of products. So if you see the different products and their effects, so tryptophan derives serotonergic metabolism in the gut-brain axis, crosstalk. What happens is, and upon absorption in the gut, L-tryptophan is metabolized through three pathways, particular to bacterial cells. That is, one is the indole pathway of gut microbiota and mammalian cells involving the kynurenine and the serotonin pathway. So tryptophan is converted to 5-hydroxytryptophan by tryptophan hydroxylase, and thereafter converted to 5-hydroxytryptophan by the enzyme aromatic L-amino acid decarboxylase. And this tryptophan is also metabolized into, as I told you, indole derivatives and kynurin, which also perform various biological functions. So, an unbalanced kynurin metabolism has been suggested to underlay development of microbiota gut-brain axis disorders. So, in normal conditions, kynureninic acid and quinonic acid do not cross the blood-brain barrier in considerable amounts, but it is permeable to kynurin, which is principally transformed into kynureninic acid in the astrocytes and quinonic acid in the microglia. And the constant intracellular calcium increase involves a cascade of dangerous events, including the activation of protein kinases, formation of ROS or the reactive nitrogen species, lipid peroxidation, leading to apoptosis or necrotic cell death. And also, this quinolic acid may participate in the pathophysiology of several psychiatric and neurodegenerative disorders. Though kynureninic acid is considered as neuroprotective, acting as an antagonist at NMDA receptors, both in the CNS and in the enteric nervous system. Now, coming to the serotonin-mediated bidirectional crosstalk between the gut microbiota and brain, serotonin plays a key role in microbiota gut-brain communication to modulate the gastrointestinal central nervous system functions. And this action is mediated through various signaling mechanisms between the 5-hydroxytryptophan receptors located in the post and the presynaptic neurons at the CNS and intestinal serotonergic neurons and in different cell types. So here the different cell types here, for example, this SCRT, microbial associated molecular patterns from the microbiota directly affect the serotonergic system, mainly through the modulating the activity and expression of serotonin transporter, SCRT, and serotonin receptors, as well as synthesis of 5-hydroxytryptophan in GI tract. And stored into vesicles forms through the vesicular monoamine transporters. Here, VMAT1 in the EC cells and VMAT2 in the neurons. Here. So, this serotonin is further released into the extracellular spaces where it binds to different serotonin receptors at the same place taken up by the neurons, enterocytes, or platelets through the SERT, limit the 5-hydroxytryptophan mediated signaling crosstalk. So, this is the mechanism of serotonin. Next is the NPY system, neuropeptide Y system, which is found at all levels of the gut-brain and the brain-gut axis, and is a major system expressing this NPY comprises the enteric neurons, primary afferent neurons, several neural pathways throughout the brain, and the sympathetic neurons. And in the digestive tract, the NPY and the PYY inhibit the gastrointestinal motility and electrolyte secretion, and in this way modify the input to the brain. And this peptide YY is also influenced by the intestinal microbiota. And this NPY, that is neuropeptide Y, exerts via stimulation of Y1 receptors a pro-inflammatory action. So the effect of NPY on gut bacteria is similarly to substance P. It is found to exhibit a direct microbial effect against various gut bacteria, including E. coli. So this is a mechanism. The hormones directly or indirectly signal to NPY, AGRP, and POMC CRT containing neurons located in the hypothalamic arcuate nucleus. And this arcuate nucleus plays a role in the regulation of appetite and energy expenditure. So the activation of NPY neurons has an orexogenic effect, whereas the activation of POMC CRT neurons has an anorexogenic effect. And this has NPY has a distinct impact on the immune function within and outside the GIT. And they release NPY released from the sympathetic nerve fibers act on Y receptors, notably Y1, Y2, Y4, and Y5 subtypes, expressed by distinct class of immune cells. And NPY acts as a paracrine or an autocrine immune mediator because immune cells themselves can produce and release NPY. Here. Then coming to the immune signaling. So here in the immune signaling, that is, the immune system reacts to what is it sees in either the gut or brain and creates cytokines. It can create cytokines, which can be either inflammatory cytokines or anti-inflammatory ones. So this is to either launch a robust immune response or to calm down and go into homeostatic mode. So this mucosal immune system, around 70 to 80% of our immune cells are in our gut, getting educated and prepared to respond in case of any potential danger. So they're highly reactive to whatever is the state of our gut. If we have a leaky gut, the lipopolysaccharides found in the microbiome could leak through the gut, but the mucosal cells are right there, which would respond to the pathogen to prevent a widespread infection. So the secretory immunoglobulin A represents the first line of defense of the mucosa, and it is a component of the adaptive immune system and is an important player to protect the epithelium. And it has an impact on the regulation of gut microbiota, and it is central to the normal function of the GI tract as an immune barrier. And this mucus layer is crucial to the process of protection and homeostasis of the gut. And it regulates, this mechanism is done by regulating the pH in the gut lumen and influences the composition of intestinal microbiota by producing biofilms to protect the good bacteria and fighting off the bad bacteria. And it also downregulates pro-inflammatory responses from highly pathogenic bacteria and allergenic antigens and promotes the retro transport of antigens and also prevents hyperstimulation of the local non-mucosal immune systems. Then lastly, if you come to the gut motility. So there is a strong correlation between the gut microbiome and how quickly food moves through the gut. And a lot of it is mostly controlled by the brain. So reduced motility has been linked to a number of diseases, and one of which is called the SIBO, that is a small intestinal bacterial overgrowth. So this small intestine, which typically does not have as much bacteria as there is in the colon, so the SIBO, that intestinal transit time, is reduced. So there is a lot of bacteria just sitting around in the small intestine. So reduced motility may reduce the biomass and diversity in the distal gut. So kind of the opposite of SIBO, too little bacteria in the small intestine. So, so just to conclude, so we know that the gut's listening is via the enteroendocrine cells, the mucosal immune system, the circulation, the vagus and all. Understanding this intricate dance of the gut and brain and the microbial world within allows for a deeper appreciation of the gut's profound influence on brain function and vice versa. So any disruption in the gut-brain axis, whether through dysbiosis, stress, or inflammation, can throw off its rhythm, impacting not just the gut function, but overall health. So recognizing the gut as an active participant in this process, rather than a passive responder, shifts our understanding of our health itself. So I hope I'm very much clear. And once again, I thank the team of Government Medical College, Medak, for inviting me to present this small information to you. Thank you. Thank you so much.
Thank you, ma'am, for sharing your expertise with us today. Your session has deepened our understanding of physiology behind the gut-brain axis.
Thank you, ma. Thank you so
Ma'am
much.
is now open to any questions from the audience, candidates online and offline. Anybody who has a query, you may please come forward.
Any questions? You can unmute and talk. Any questions from online side? Jaya, ma'am.
Dr.R.Anitha. This is Dr. Jaya. Dr.R.Anitha, can you hear me?
Hello, Jaya. How are you? Thank you.
Good, Dr.R.Anitha. You have given us such a wonderful session. You have opened the session so well. Dr. Shanta Madam has given us such a good session and you have opened it so well. We have
Thank you,
learned.
Jaya. Thank you so much.
We have learned so much from you, Dr. Anita. I have one question for you. You were talking so much about everything, all the physiology, but I want one biochemistry question from you.
That I think you can answer better.
So you were telling about the microbiota. You were telling how it improves the gut system and how it improves the nutrition aspect as well. So
Yeah.
I just wanted to congratulate you rather than question you.
Thank you so much.
Thank you, Dr. Anita.
Thank you, Jaya, for your concern. I think the next sessions will give you answers to all who have questions in their mind. The next few sessions will be giving you answers.
With that